Hello friends, this is Rashmi Trivedi. I would like to give you a short glimpse of an outlook to drug discovery. Let us see how a typical new drug travels from laboratory to the patient. The first step in the drug discovery process is a target selection, where you identify the receptors, and these targets or receptors are studied through genomics, proteomics and bioinformatics. Once you have studied the target in detail, the next we can begin with the lead discovery, where a new drug molecule is synthesized and isolated. Also, along with the synthesis of a new drug, your drug can be from the natural origin, either from a plant or animal. And once you have identified the molecule, you have to develop a assay for it, which will be further used for identification of the drug. Once the new drug molecule has been synthesized, the medicinal chemistry department develops a laboratory for the developed molecules, where several analogues of the molecule which has been discovered are made. Out of this several molecules, the best ones are selected and further they are carried for the in vitro studies. In vitro studies, drug affinity and selectivity is tested and cellular models are chosen. Also, the mechanism of action are studied out in the laboratory. In vitro studies are also called as in vitro because they are done in laboratories outside the bodies. Once the in vitro studies are successful, next we can move to the in vivo studies, where the animal models of disease states are chosen and the mechanism of action is checked into animals. Further, we go for clinical trials and therapeutics. Here in next slide you can see that the drug discovery process has been classified into two classes, that is the preclinical studies and the clinical studies. First we come to preclinical studies, that which we have discussed in the earlier slide. A research team is formed and objectives are set. This research team then aims and develops a new drug molecule. New drug molecule is then tested for the efficacy and safety in test tubes and animals, and the best molecule is further carried out for the formulation. When you talk about the formulation is transformation of a drug molecule into the dosage form, which are tablet, syrup or other dosage form. Once the formulation is developed, we go for stability and scale-up studies, and after the safety has been studied in the animals, now the pharmaceutical companies files an investigational new drug application with the Food and Drug Administration, which we called as FDA. Once the FDA approves this IND application, the clinical studies begin. The first phase of the clinical study is phase 1 studies, in which the drug molecule is studied for the tolerance in the healthy humans, and if they are found to be safe, and in the phase 1 studies the drug molecule is successful to move towards the phase 2 studies. In the phase 2 studies, the efficacy of a drug is checked into the patients. Once the phase 2 is successful, the drug molecule then moves to the phase 3 studies, where the clinical trials are done in the n number of patients. The number of patients is more than that what was carried out in the phase 2 clinical trials. Once phase 1, phase 2 and phase 3 are cleared by a drug molecule, now a pharmaceutical company can file a new drug application, and this application is reviewed by the FDA. All the results and all the analyses are checked, and then the FDA gives a green signal and the drug is approved for marketing. So this is a journey of a drug molecule from the laboratory to the market, and this is how the drug reaches to the patient. Here we have discussed we have seen a drug molecule. We know that it is a pharmacologically active ingredient in your tablet or the syrups. We all know that there are various dosage forms. Here I have discussed them. These are the solid, semi-solid, liquid and gases. Under solid comes the unit solid dosage form, which are tablets and capsules. Bulk dosage form, which are the powders and dusting powder. Semi-solids are creams, paste, gel and suppositories. And liquid, syrup, solution, emulsion and suspension. Emulsion and suspension are the biphasic liquid dosage form, and the syrup and solutions are the monophasic liquid dosage form. We also have another class which is the gas. Examples are the inhalers and aerosols. One which are used in asthma and other conditions are the aerosols. In the next slide, we can see a typical tablet manufacturing process where there are two mechanisms, wet granulation and the dry granulation. And in the dry granulation, we do not make because the drugs are sensitive towards the moisture, we go for the dry granulation. Granules are formed, and at the end, through the tablet press, you get a tablet. While in the wet granulation, we use the wet granules where you can use a water as a binder or any other solvent as a binder. So along with the tablets and the classical examples of the dosage form, we have a new drug delivery system which include microemulsions, liposomes, solid lipid nanoparticles, iontophoresis, submicron emulsions, ethosomes, solid dispersions, and microspheres. All these are called as new drug delivery systems because they are having several more advantages than the classical one. They are mostly used for the controlled release and the target release. And so this was all about what kind of dosage forms we have. And at the end, I would like to tell you that Indian pharmaceutical industry is the third largest in world volume wise. 40% of the total production which is done in India is exported. With the market value of $2 billion US, it contributes to the 16% of total Indian economy. And not only this, it also satisfies 55% of US and 40% of Europe's drug demand. So I hope you have got a glimpse towards a drug and dosage form and the contribution of Indian pharmaceutical industry. Thank you.